let's continue where we left in the last class. We were discussing different types of ligand and their forms, their composition, and how these ligands bind to their receptor depending on their nature, whether it is abiotic, biotic, or whether they are soluble, insoluble, or they are present as a composition which is hydrophobic or hydrophilic. Now let's understand the signaling in context of signal. What I mean by that signals are around us. I gave in the example in the previous class that a cell is surrounded by a milieu, an environment which is rich in signal. So, and also we concluded that if there is a cell which has multiple different signals which respond through multiple distinct receptors, then there is no issue. So now let's see how cell behaves when there are multiple of these signals come together. So we can understand that a signal, when it comes in combination, can elicit very distinct response in the cell. In one of the earlier classes we saw that how a cell needs to divide, a cell needs to differentiate, a cell needs to induce a defense activity, right? These are different acts being carried out by same cell. So the signal which is present around, or the combination of signals which is present around, that will guide which kind of response happens. So we can simply understand this as many to one kind of signaling. Here many is signal, very different types of signal, and the response here is more in terms of receptor. Many cells, many signals will come, they will bind to the cell, and there could be one receptor through which different responses are generated. Let's see, for example, think about a cell which is present in environment and it is continuously getting these signals which are called XYZ. This is the basic minimum signal which is always present, and the purpose of the signal is to ensure that cell survives whatever environment it is present in. It homeostatically grows without showing any problem, and an equilibrium with the environment is maintained. Such situation could also be important for cells which undergo renewal, or I should say self-renewal. Some unipotent cell, they keep dividing and making sure they are generating themselves. So such basic signals like XYZ mentioned here is required for survival and the renewal. Now let's think about scenarios where more signals are present, which means that XYZ are always present and additional signal comes. This additional signal will guide what is the fate at the end of signaling. Now think about this scenario where there is a cell which could be a precursor cell. Remember I said that self-renewal is going to happen in the previous example. So here those XYZ signal were sufficient for self-renewal. But the presence of the signal D now asks this cell to differentiate, and this new cell has identity which is different from the precursor cell. It is more differentiated cell. Now yesterday we were thinking about immune cells, a population of immune cells which will be having macrophage also, neutrophils also, B cells also, T cells also, right? So where does this population come from? These B, T cells or macrophage and neutrophils, they are differentiated form of cells. They probably can come from a very early precursor, and a specific differentiation signal allows them to go from a precursor to B cell or T cell or neutrophils. So what is important to remember here is that there is one set of basic signal which is required for renewal and survival. Then addition of a signal guides which way the process will move, and this is achieved by a unique signaling which is responding to the presence of this new signal, which is D. Let's take another scenario where cell need not differentiate, but rather it has to grow in size, it has to duplicate. Yesterday we discussed this, that why do we need signaling? Signaling is required for duplication and proliferation and growth. Same thing is here. Now you think about a scenario where the same cell is in an environment which is receiving this X, Y and Z signal. Remember, this is important for cell survival or renewal, but now it has to increase in number, right? So you can consider that in case of precursor cell or a unipotent cell which has to keep generating itself, we saw renewal signal. But if it has to increase in number by dividing, or it has to just grow in dimension before it can divide, so you get additional signal in form of G and P for growth and proliferation. So once this cell grows, it gets a signal that it has to proliferate, and that's where it divides and gives rise to daughter cells. So what you understood, that here a set of combination of signal which is always present is required for survival, but additional signal which is coming, what is the nature of the signal, what is the source of the signal, what is meant from the signal, leads to a different process altogether, whether it is differentiation, survival, renewal or proliferation. Now you can think about a scenario where no signal is present. You should also ponder that, is there a condition when cell has no signal? I just told in previous classes that cell environment is rich in signals. So how can we talk about no signal, right? So in this case, no signal means those X, Y and Z signal which were important for survival. So when there is no signal X, Y, Z type, you do not have survival input, and that's where the cell can undergo death or necrosis. So this is one scenario where in absence of relevant survival signal, cells undergo death. But you can think about another scenario where X, Y and Z signals are there, are present, but there is an overwhelming toxic signal, because of which it will go to the death. Remember, I told in previous class, generally there is no contradictory signal, because if X, Y and Z are survival signal, then presence of toxic signal or death signal does not make any sense. That is why I said, for whatever reason, a conditional situation where there is abundance of toxic signal, this will lead to the death kind of scenario for the cell. Now there could be a situation where one signal, or I should say same signal, when I say same signal, take any scenario for that matter, for suppose I say a neurotransmitter. The same neurotransmitter, I gave you example of serotonin and dopamine in previous class. The same neurotransmitter can evoke different responses in different target cells. So this will be an example of one to many responses, where same signal is eliciting different responses. If we can understand the statement of one signal to many responses also in a scenario where one cell which is receiving this unique signal, but depending on the condition, it will respond differently. In previous slides, we saw that how same cell could be responding differently depending on the signal. But here the scenario is that the cells are going to respond very differently to same signal. Let's understand that in little more detail. A basic signaling event which will be happening. The signal is present on this environment, and then you have a receptor to which this signal binds. by just representation you can understand this is more like a hydrophilic signal which cannot pass the membrane. So now this binding initiates some changes in the receptor, and this change must be understood by these proteins which are present as a signaling protein. Because whatever signal is present outside, it must convey the right message inside. In the very first slide I told you in the first lecture that signal must be comprehended and it should convey the message it has. So the signal which is present outside, it binds to the receptor. The only way to convey message is to bring about some change in the receptor. That change is interpreted further by these signaling proteins. They could be present in a cascade or they could be present independently. Now in this case, if you think that this is a linear cascade, you will have protein 1 getting activated, which will send signal to protein 2, which will send signal to protein 3. Now in this scenario, all these 3 proteins have to be activated in a given manner. Now final molecule which is giving the signal out is this molecule. So this will be directing the signaling depending on the nature of the signal. There could be related signals, like I said, CO, NO, CO2. They look very similar, gaseous molecules, but their signaling will meet its destination depending on which signal was that. So what simply is explained here is that the final molecule, which is this in the cascade, can drive the signaling in a given direction. Like consider a scenario where in response to signal we saw chemoattraction. Remember that this dictyostelium was going towards the cyclic AMP zone, right? So for moving towards a location or to particular point, you need lot of movement inside the cell. The movement of or dynamics of the cytoskeleton has to be affected. If that is desired from the signaling inside the cell, you will have cytoskeletal changes which will change the morphology and will initiate movement. Sometimes you need to activate an enzyme which will work and affect the metabolic activity, right? Suppose in response to what you have eaten, the system has to be activated and proteins have to be digested, right? So the system will be going in a direction where enzymes are activated and the metabolic activity is taken into effect. Third option is that, which is often seen in many cases of signaling, is that the final goal of signaling is not to induce a movement in cell or not to perturb directly or immediately the metabolic activity. Rather, it feeds in a system which prepares the cell for better responses later, where you need to induce some sort of activation of transcription. So in response to the signaling, this molecule will allow the gene expression. Gene expression in the sense that particular proteins will be made from their transcript, which will help facilitate the eventual goal of the signaling. Think about a scenario where there is high glucose in the system. You really need to remove that glucose from blood circulation and store in the cells, right? In that case, you will allow the transcription of things which will activate and promote taking up of glucose from the blood circulation. So eventually, one signal can allow different kind of processes or signaling inside the cell. Receptor could be same, signal could be same, just that cell type could be different, and that cell type will take the signaling finally in direction A, if I can say, B or C. So this is kind of one-to-many example which we see here. So let's take an example of this, what I just told you, that single signal molecule. Consider this neurotransmitter I told you, acetylcholine, which is supposed to bind and activate many processes inside the cell. So think about a cell which is cardiac muscle cell. It has the receptor. Since we know the signal, we also can extrapolate and say what is the receptor. This is an acetylcholine receptor. So in presence of acetylcholine, then binding of acetylcholine happens with the receptor. And what is the end result of that? You will see that the heartbeat rate is affected. Molecule is same, the target cell is different, receptor is same, and you see a heartbeat rate difference. Now think about a scenario where you have a salivary gland cells. Again, the receptor is same, which is acetylcholine receptor. The ligand is same, which is acetylcholine. Now what happens in response to binding of this acetylcholine to its receptor here, you would notice that these granules are secreted. Even similar situation you will find in the neuromuscular junction, where vesicles filled with acetylcholine, they fuse and release their content into the synaptic cleft. Similar thing is happening here. However, here rather than acetylcholine filled vesicle, here there are vesicles which are filled with enzymes, and acetylcholine itself is serving as a signal. So binding of acetylcholine to the salivary gland cell induces fusion of these vesicles which you see here to the membrane, and then release of the content. Basically this is what will constitute the saliva and will activate the process of digestion. So if you compare with the previous situation where we saw acetylcholine binding to its receptor which is present on the cardiac muscle cell, versus this acetylcholine receptor which is present on the salivary gland cell, you saw that the processes are very different eventually, which are meeting as an end goal of signaling. In one case you saw heartbeat rate decreases. Here you see that vesicle filled bags fuse to the membrane and release the secretion to form the saliva. Now the third scenario with the same acetylcholine molecule is with the skeletal muscles. This acetylcholine, when binds to a skeletal muscle cell, so through its receptor, it allows the contraction of the skeletal muscle. So all the movement which we can perform along our bone joints using skeletal muscle, whether it is a simpler movement or a much bigger movement, is because of the acetylcholine binding to its receptor on this skeletal muscle fiber cell and allowing the contraction to happen. In one of the previous classes, I showed you a GIF where you saw that how acetylcholine was being released on the muscle cell surface, skeletal muscle cell surface, and it was inducing the contraction of the muscle. So this basically represents a situation where you have single signal. Receptor is also same, just that the cell type is varying. Depending on the cell type, you will have different responses. So why you should have different responses to same signal and same receptor? This is what exactly I told you here, that the signal will be directed into one of the given direction, right? So if it is required for cell to move or contract, you will probably see something like this happening. When it is required for enzymatic activity, the saliva and the salivary gland which you see, there you will probably use an enzyme activity. So downstream to a signal, which is in this case acetylcholine, binding of acetylcholine to its receptor, which is here acetylcholine receptor, you will see different activities being performed. So what we can say that in this class we understood that the presence of signal or a combination of signal drives the response, right? Like we saw, there could be a survival signal which is present always. Then you need another signal so that cell can grow and divide. Maybe you need a signal where cell needs to differentiate, or you, for any condition which is present around the cell, absence of these signal can lead to death. What you also understood that signaling output to a particular situation could vary depending on what is the target cell, right? We saw example of cardiac muscle, salivary gland cells, and the skeletal muscle cells. Right? So also one more thing which you understood, that inside the cell, in response to binding of the receptor to the signal, depending on the signaling proteins and the requirement of that cell, signaling is diverted to meet its goal, whether it is about the movement of the cell, whether it is about activating an enzyme which to carry out a metabolic process, or ensuring that gene expression happens and that meets the goal. In summary, a signal can drive different responses in different cell types, and combination of signals to a given cell type will drive different end result like growth, proliferation, renewal, survival or death. Thank you.